All right, welcome everyone. Thank you very much for joining us this evening, morning or afternoons, depending where you are. We just wanted to welcome to our first Single Cell Ninja seminar together with Vizgen. Today, this seminar series is essentially, we want to show you these technologies that we think will change the way that we understand biology or will help us understand biology a little bit better. And today is the time of Murfish, and it's my pleasure to introduce to you Dr. George Emanuel. George is the scientific co-founder of and director of technologies and partnership at Vizgen, and he's a trained biophysicist at Harvard University from the laboratory of Dr. Xiao Shang. And he spent the past decade developing super hardcore technologies, and one of those is high multiplex RNA fluorescence in situ hybridization technologies. And perhaps the most amazing legacy that he has given to us is the invention of the high throughput image-based screening technology and demonstrating the capacity of the Murf technology. So within Vizgen, George is in charge of directing the technology development of the Merscope Platform, as well as coordinating the strategy of partnerships to enable researchers to apply the Murf technologies to their research. So in this presentation, George will introduce you to Merscope. I'm sure that you all are excited to know how it works and what it does and what it can do, which is essentially an end-to-end in situ genomic platform to enable the direct profiling of the spatial organization of intact tissues and with a scale which is genome-wide. So a few housekeeping notices here. So this presentation is going to be recorded. So for those that didn't manage to attend today, don't worry. We will have a recording on our website, on YouTube. I'm pretty sure we will make it available on Twitter. And we will, if you have any questions along the way, feel free to put it on the chat, and we will start, selecting a few of them to present it to George at the end of the presentation. And without further ado, George, this is your turn to tell us a little bit about the Murf.
Great, thanks for the introduction, and I'm really excited to be here today to introduce some of the work that we've been doing at Vizgen over the last year and a half as we work towards putting together the complete solution for Murf technology, which we call the Merscope solution. So today, before I get started, I just want to lay the framework for how we see this sorts of spatial genomics technology fitting into the broader landscape of genomics technologies, because maybe 10 or 20 years ago, we really saw this new level of understanding emerge through the widespread adoption of bulk sequencing through these next generation technologies such as Illumina and such. So with this technology, you're able to grind up a whole tissue, and then by putting it through the sequencer, you're able to count how many of each different RNA species was present across that tissue. But although this lets you understand sort of which genes might be important across different tissues or across different treatment conditions, it fails to reveal two important aspects of biology. So first, these biological systems are comprised of individual cells. When you look at the bulk expression across the whole tissue, you don't understand what each individual cell might be doing. Second, you lose the spatial information because cells don't just function in isolated droplets. Rather, they're very actively communicating both with each other and the broader surrounding environment. So with bulk sequencing, by grinding up the tissue, you also lose all of the spatial information. More recently, within the last 6 to 8 years, we've started to see the adoption of single cell sequencing based technologies. So here, often cells are sorted into a multi-well plate or into droplets. And then by using sequencing, we're able to count how many of each transcript appears within each of the cells. So we're able to get the transcriptome out at a single cell level. So this has provided a lot of insight over the last few years into the cellular composition of these different biological systems helps us discover new cell types and also understand how these different cell types are changing in different biological conditions, whether it's a disease or some sort of treatment towards that disease. But with single cell sequencing, you still have to dissociate the cells, so you still lose this spatial organization. But with this next generation of genomics technologies, these spatial genomics technologies, where you can directly interrogate the molecular composition of an intact tissue slice, now you're able to do this very highly multiplexed measurement that's on the scale of hundreds or thousands of genes, while maintaining the exact spatial location of each of those genes. So this really enables you to do sort of single molecular, single cell, spatially resolved transcriptomics. So at the same time, you can map out the cellular composition of a sample. Not only do you know which cell types are there, but you know exactly where each of the different cell types is present within the tissue, which gives you this extra level of information about the possible biological function of the different cell types that you identify. So as we look towards the future and looks towards the adoption of this spatial genomics technologies, we're really excited to start with our commercial launch of the Merscope Platform. So the Merscope Platform is the end-to-end solution that enables Murfish technology. But through the course of this talk, I'll first discuss Murfish technology in more detail. I'll describe how we're able to achieve this single molecule, high resolution, spatially resolved transcriptomic measurement. From there, I'll go into more details about the Merscope solution, which is how we really hope to streamline the process of running a Murfish measurement within whichever research facility could benefit from Murfish technology. And finally, I'll go into some biological showcases, some demonstrations of what sort of insight Murfish technology might be able to provide to different biological questions. But just to start out, I'll introduce Murfish. Murfish is this highly multiplexed, high-resolution spatial genomics technology. And as with any technology, it builds upon what was there before it. And in that case, this is a single molecule fish. So in single molecule fish, you design probes, fluorescently labeled probes, that are able to bind specifically to a target transcript. So then once you incubate these probes with the sample, these probes stain the exact transcript that they're designed to target. And then when you look at the sample under a high resolution fluorescence microscope, then each copy of that RNA that you're targeting shows up as a single diffraction limited spot under the microscope. For example, the image that you see on the right. So here, each of the different points corresponds with one copy of that transcript. So then by counting up the number of spots that you see, you can quantify the expression of this gene across this cell, and then by imaging larger areas, you're able to quantify the expression of the gene across the whole tissue. So it's a very quantitative approach by directly counting the RNA transcripts within the tissue where they're being expressed in. But as with many traditional fluorescence technologies, single molecule fish is traditionally limited to just being able to measure a few genes at once, just because of the spectral overlap in the different fluorescent dyes that you're able to use. So because of that, Murfish is able to greatly expand the multiplexing capacity of single molecule fish by, rather than measuring one gene at a time, we assign each of the genes a binary barcode, and then we read out that binary barcode using sequential rounds of single molecule fish. So for example, in the first round of single molecule fish, we stain the sample in such a way that all of the genes that are assigned a binary barcode that contains a 1 for the first bit position show up as the single molecule fish spots, while all of the genes that were assigned a barcode that contain a 0 for the first bit position show up, remain dark, so they don't show up as single molecule fish spots. So this allows us, by the pattern of single molecule fish spots, read out the first bit in the binary barcode. Then we're able to extinguish that signal and then restain for the second bit in the binary barcode. And now we get a new pattern of single molecule fish spots. Now it corresponds with all of the barcodes all of the genes that were assigned a barcode that contains 1 in the second bit position. So again, we image this pattern of single molecule fish spots to read out the second bit, and then we continue this process in order to read out the full binary barcode, the third bit, all the way up through either the 16th bit if it's a measurement on the scale of 100 genes, or the 32nd bit if it's a measurement on the scale of 1,000 genes, or even the 70th bit if it's a measurement on the scale of 10,000 genes. But then from these binary barcodes that we read out, we can look at the pattern of brightness and darkness across each point across the tissue in order to identify the binary barcode that is present at that point in the tissue. And then by identifying the barcode, we know exactly which gene is there. So this enables the very highly multiplexed spatial transcriptomics measurement. But it's also very high resolution, as you can see. We're detecting the individual RNA transcripts, so we know exactly where each of the hundreds or thousands of transcripts that are from the genes, from the hundreds or thousands of genes that we probed, where the transcripts are within the sample. So by combining this combinatorial labeling scheme, this binary encoding scheme, with the sequential imaging to read out these binary barcodes, along with error-robust barcoding, we're really able to achieve this very high resolution, very high accuracy spatial transcriptomics measurement. So the technology itself was originally developed back in 2015 in Xiaowei Zhuang's lab. Since then, there's been a number of additional publications, both coming from her lab, but now new publications starting to emerge from the broader community, where people have started to apply this Murf technology to a whole range of different research questions. In addition, we also see a growing number of institutions starting to adopt Murf technology. These are both through home-built systems where they build up the whole workflow on their own, but also a growing number of institutions, some that are not even listed on the slide, that are enabled by Vizgen to really run this Murf technology to answer their own biological questions. But from the way that the technology works, there are just a couple of advantages of Murf technology that I want to briefly highlight, that I'll go into a little bit more detail on later with some of the biological showcases. But first, since Murf is an imaging-based technology, we're able to capture more cells across the sample just by tiling the XY stage across additional regions of the sample. So in a typical measurement, we're able to measure up to a square centimeter of tissue and detect hundreds of thousands of cells across that square centimeter of tissue. So there's very high cell throughput per sample. Next, we have this it's a very flexible measurement. Murf works on pretty much any sample where single molecule RNA fish works. And we've demonstrated that Murf works successfully on a broad range of different mouse and human tissues. Next, Murf is very sensitive. Since single molecule fish is in many cases considered a gold standard for quantifying RNA expression, since Murf builds upon single molecule fish, we're able to maintain that very high sensitivity. Next, since Murf is a single molecule imaging technology, we have very high resolution. We know exactly where each individual transcript was detected. So then that enables us to not just do analysis on the intracellular distribution of the RNAs, but also the group RNA transcripts that are being expressed within the same cell in order to enable single cell level analysis. And then finally, because of the binary encoding scheme, we're able to adjust the length of easily adjust the length of the barcode in order to adapt to the throughput, the gene throughput that's required for any sort of measurement. So we can multiplex anywhere from Murf as a technology can multiplex anywhere from tens to hundreds to even thousands of genes that are being measured at the same time within the same sample. So from these advantages, I think as we work towards launching the Merscope platform, we're really excited about the platform because it is this genomics tool. And as you see with bulk sequencing, as you see with single cell sequencing, these genomics tools can be really applied to a very broad range of different applications. So just to highlight a few that we're we're very interested in. I have a couple highlighted on this slide. For example, neurodegeneration. And with neurodegeneration, being able to understand both where the different plaques that are building up with these different neurodegenerative processes and how the different cells are responding in the vicinity of those plaques, I think can give new insight into the progression of these different neurodegenerative diseases. Similarly, with cancer immunotherapy, being able to understand the immune landscape, sort of which immune cells are at which position in the tumor and what is their activation state, I think can give new insight into the progression of different cancers and also guide the development of new treatments. And then finally, with infectious diseases, there's always this sort of pathogen host interaction where there's certain cells that are infected with the pathogen, and then understanding how those cells are either evading the immune system or how the host immune system is working to fight the infection. I think there, since the infected cells have some spatial distribution, being able to do a spatial measurement to understand how the immune or how the host system is responding in the vicinity of these infected cells, I think can also give a lot of insight. So this is just a couple examples, but we do imagine that MERFish can have a very broad range of different applications. So that's why we're very excited to launch the Merscope platform, which is this complete workflow solution to enable MERFish measurements. So it seems like every day here at Vizgen, we either have a new instrument coming in or we have an instrument going out. So currently we have a number of instruments in-house that we're just finalizing some of the different components to make sure that they can produce robust measurements as we approach our commercial launch later in the summer. But from this Merscope solution, you can see there's these two boxes. There's the box on the left that contains the laser, the computer, and some additional electronics. And then there's the box on the right that contains a microscope, a fluidic system that is able to perform the MERFish imaging and staining steps automatically. So in a single measurement with the Merscope, we're able to image up to a square centimeter of tissue with this single molecule resolution across the whole tissue slice. With the solution that I'll describe, we're really aiming to combine the advantages of MERFish technology, which is this very high sensitivity and flexibility that comes from the nature of it being based on single molecule fish measurements. We have this high multiplexing capacity that comes from the binary encoding scheme. And then we also have this uncompromised accuracy that comes from the way that we choose the binary barcodes that we assign to each of the genes. So we want to combine the advantages of this chemistry with automated, easy to use hardware solution, and then additional reagent and software tools to facilitate the whole process. So from here, I'll describe what the process actually looks like all the way from designing a gene panel. So our default offering with the Merscope solution is an entirely custom gene panel where you're able to specify whichever genes that you want, and then Vizgen will produce a probe set that then you can use to stain your sample. So the sample staining process, we typically currently you start from fresh frozen tissues, but we're also have had success with fixed frozen tissues and some initial success with FFP prepared tissues. But from a fresh frozen tissue block, we slice it into a 10 micron thick tissue slice. We adhere that slice onto our Merscope slide. And then there's just a few steps in order to prepare that tissue slice on the Merscope slide to be put onto the instrument. So first, we fix and permeabilize the sample just to hold everything in place and to allow the probes to get into the cells to stain the RNA. From there, we have a cell boundary staining kit that helps with segmentation in some of these denser tissues. But after the cell boundary staining, then we perform hybridization. For hybridization, we just put a drop of this ready-to-use aliquot of the encoding probe mixture onto the sample, and then we incubate it for a couple of days to let the encoding probes bind to their respective RNA targets. Then once these probes are hybridized, then we have a gel embedding and clearing process in order to clear away the proteins and lipids that could cause autofluorescence background that could degrade the quality of the measurement. So since there's in different tissues, different cell systems, there's all sorts of different aspects or different components that could contribute to the autofluorescence background. So then by including this process, we're able to remove a lot of that fluorescence and autofluorescence from the sample and get very clean images. So after the sample is prepared on the bench, which typically takes 4 or 5 days, but is only a couple hours of hands-on time, then the sample is ready to go into the Merscope instrument. So we assemble the sample into our fluidic chamber and then place the fluidic chamber onto the instrument along with the imaging reagent cartridge. So the reagent cartridge also goes onto the instrument, and it contains the different hybridization buffers for staining each of the bits in the binary barcode. So then after loading the instrument, then it's able to run through the whole Murfish measurement automatically in order to acquire all of the images, and then it also processes all of the raw images in order to get the output that's most useful for downstream biological analysis. So that includes these 5 output file types. So first is the list of all of the transcripts detected within the sample. So this is a CSV file. So we aim to make all of these different output files very easily accessible or easily importable into different sorts of downstream tools. So the first is the list of all of the detected transcripts. So this could be a list of tens or even hundreds of millions of transcripts for a single measurement. And then for each transcript that was detected, it includes the transcript name and then the XYZ coordinates of that transcript within the tissue. Next, in addition to the Murfish images, we also acquire images of DAPI and poly-T stains. And then from these images, to help facilitate downstream image processing analysis, we stitch them into these very large high-resolution mosaic images. Then the next 3 outputs all relate to single cell analysis. So with each sample that goes through, we also segment the cells within that sample. And from the cell segmentation, we're able to construct the transcripts per cell matrix just by counting the number of transcripts that appear within each of the segmented cell boundaries. So this transcripts per cell matrix is very similar to the transcripts per cell matrix that you'd see from a single cell sequencing measurement. And as such, you can fit it into the same sorts of tools like Seurat or ScanPy in order to perform clustering or differential expression analysis to understand what sorts of cells are there within your sample, and then what are the key marker genes that are defining the different cell populations. But then since it's a spatial measurement, we also have the cell metadata output, and the cell metadata includes the spatial information about each of these cells, for example, its volume, and then its XY coordinates. So this allows you to perform spatial analysis on the organization of the cells within your tissue slice. And then finally, we also export the full cell boundary polygons as this HDF5 file so that you can see exactly where the segmentation algorithm drew the boundaries for each of the cells. So we aim to make these different files as open as possible, but we also have a visualization tool that lets the user interactively explore the full data set. So it shows the transcripts, it shows the images, and it also shows the cells and lets you color the different components by whichever feature you want to really start to understand the biology across your sample. So from there, I think what's really important about the Merscope solution is that it enables new biological discoveries. So I do want to go into a couple of biological demonstrations of Murfish technology, both some that were developed within Vizgen, where we've been running the measurement on a very large number of samples over the past 1.5 years since getting Vizgen started. But I also will briefly highlight some of the demonstrations from the published literature as well. So the first example that I want to go through is this mouse brain receptor map that we want put together within Vizgen. So here we selected a panel of 483 genes, and then we measured that panel across these 9 mouse chronoslices. So the panel contained in all 483 genes, but that contained about 50 or so canonical brain cell type markers, and then about 430 or so GPCRs and RTKs. So the aim for this measurement was to map out the spatial organization of these GPCRs and the RTKs in the context of which cell type they're being expressed in. So for this, we actually publicly released this data set. So if you're interested in seeing exactly how Murfish data looks on your own, then you can go to the website and find where to download this data set. And then we have the full Merscope output for each of these 9 slices. So you can start to understand what Merscope output looks like in the context of real measurement. But from this measurement, I do want to go back to some of the Murfish advantages that I was highlighting earlier and how they fit in the context of this measurement. So I'll focus on this one chronoslice. So here we're just showing 8 of the 483 genes just for visual clarity. But since we have single molecule resolution across the whole tissue slice, we can zoom into any region of the measurement and really see exactly where each of the different transcripts was being detected. So here on the zoom in for this part of the mouse cortex, we're plotting out each transcript. So you can see these dense regions along with the sparser regions in between. So the dense regions correspond with the cell bodies of the neurons. And then the regions in between correspond with the different projections coming out of the neurons, for example, the axons and dendrites. So these axons and dendrites do still have RNA transcripts in them, as you can see, but not as densely packed as closer to the nucleus. So from this high resolution, you can also see that we draw the cell boundaries around each of the cell bodies, which then enables us to perform single cell level analysis and classify these different cell types and states, as I'll touch on a little bit later. But as with any genomics measurement, it should both be highly quantitative and highly reproducible. So when we compare the Murfish output to the output from bulk sequencing, for example, we find that the genes that were highly detected with bulk sequencing that have high FPCAM are also the genes that are most frequently detected with Murfish, and then vice versa for the lowly expressed genes. So this indicates that Murfish is a very highly quantitative measurement because it agrees with other measurement technologies that are already very well established. Similarly, we find that Murfish is highly reproducible when we compare 2 Murfish measurements to themselves, to each other. We find that there's very good correlation in the copy number per cell detected for each of the genes. So with this, Murfish is both quantitative and reproducible. But the other 2 aspects that I want to highlight are the high detection efficiency, since that's really critical for understanding biology, especially at a single cell level. And then I also want to highlight the high resolution of Murfish and then what that enables in terms of understanding what each cell might be doing. So for both of these, it's easiest to discuss the advantages relative to what else is available on the spatial transcriptomics market. So here we have a matched Merscope sample with a sample measured with oligo array-based platform. So here, if we count the number of copies of each of the 483 genes detected across these matched samples, we find that with Merscope, we systematically detect about 70-fold more transcripts than detected with the oligo array-based platform. So since transcriptomics is already a relatively sparse signal, for a lot of very functionally relevant genes, they're just expressed at the level of a few tens of copies per cell. When you divide that tens of copies per cell by 70, oftentimes you're just left with maybe 0 or 1 count per cell, which really hides a lot of the interesting biology. So we can see that if we try to plot out the spatial distribution of opioid receptor delta 1. So when we plot out that spatial organization with Murfish, we get this very rich, intricate spatial organization across this portion of the mouse brain. But then if we plot out the expression detected with the oligo array-based platform, we only get a few oligo grid spots where just maybe 1 or 2 copies of this transcript was detected. So this spatial organization of the gene is completely hidden. So that's what I mean by high detection efficiency. By being able to detect nearly all of the expressed transcripts in the sample, we're able to really reveal more of the biology than was possible with currently commercially available technologies. The other aspect that we can highlight in this comparison is the resolution advantage. So again, with the Murfish measurement, we can zoom into any region across this portion of the mouse brain to see exactly where each transcript is being expressed. So if we zoom into this region, for example, we can see that here the opioid receptor delta 1 transcripts are plotted in green, and every other detected transcript is plotted in gray, just to give you some context for the brain region. But here, just by looking at where the green spots are, you can see that it's very highly localized within individual cells in this region of the brain. But with the oligo array-based platform, not only is there a lot of empty space between the oligo grid spots, but within each grid spot, we only get a single measurement for the expression level of this gene. So then from that single measurement, we don't know whether it's homogeneous. This gene is homogeneously expressed across this grid spot, or whether it is in fact highly localized within individual cells within the grid. So that's what this single molecule resolution enables, and it enables the single cell level analysis. But from the single cell level analysis, you can do lots of things that you can do with single cell sequencing. For example, based on the gene expression for each of the cells, you're able to cluster the different cells into different cell types or different cell states. But since it's a spatial measurement, then you're able to map each of those different cell types and cell states back to its exact spatial position within the tissue. So to highlight what advantage this single cell resolution has, we can look at what it means for understanding the spatial organization of different transcripts across the brain. So for example, we can just focus on this region of the cortex for this coronal slice, and we can just coarsely classify the cells. We can classify them as either neurons or non-neuronal neurons, just for the simplicity of this demonstration. But if we look at the expression of a gene across this region of the cortex, for example, this GPRC5B gene, if we look at all of the cells in this region of the cortex, we can see that this GPRC5B gene is expressed, sort of has two regions of high density expression. One's deep in the cortex, which is towards the left-hand side of this plot, and then one's towards the middle of the cortex. So without the single cell resolution, that's really all we would be able to say about the expression pattern of this gene in this region of the brain. But with the single cell resolution, we're able to deconvolve the signal between the signal that comes from the neuronal expression and the signal that comes from the non-neuronal expression. So for example, if we just plot out the expression of GPRC5B within the cells that were identified as neurons, we can see that that's really contributing to this peak of high expression that's towards the middle of the cortex. But if we look at the expression within the non-neuronal cells, then we see that this peak deeper in the cortex is really contributed to by the non-neuronal cells. So with this ability to deconvolve the signal between among different cell types, then we're really able to understand the spatial organization of the gene expression in a cell type dependent manner. So another example that I just want to briefly highlight that we've been working on putting together within Vizgen is that Murfish also works on clinically relevant samples. So here we performed a measurement of 417 genes across this mouse. This is actually human colon cancer slice. So here again for visual clarity, on the left-hand side we're just plotting out 10 of these genes, but you can already start to see the rich spatial organization that the Murfish technology starts to reveal. So the intestine is towards the bottom left of this tumor slice, and then the tumor is growing out towards the top left. So you can see that towards the bottom left of the tumor, there's lots of immune cells that seem to be infiltrating from the intestine. So in particular, here we have IgHA1, which is a B cell marker. And you see there's these large clusters of B cells that are towards this region of the tumor. While towards the other side of the tumor, there's very few immune cells present. But then, as with the brain, we can zoom into any region across this this tumor slice and see really what's happening at the tumor microenvironment level. So here we're zooming into this region where we have this large population of B cells, and we can go in a level farther and see sort of what other genes are being expressed among these cells, sort of what is their cell state. But then we can also look at how the neighborhood of cancer cells it might be different in the vicinity of this population of B cells. For example, we have this population of proliferating tumor cells that are encircling this SPP1 expressing cell. So there's already rich spatial heterogeneity at the tumor microenvironment level that we're really excited to see how researchers can start applying this to really understand how the progression of tumors and also understand what might be the best options for treatment. So in addition to looking at the spatial expression of these few genes in this example, we can also identify the different immune cell types and their distribution across the tumor. So for example, here we were able to cluster and identify the different T cells, the different macrophages and such, and really plot out in order to understand their spatial organization across this colon cancer slice. So from here, the next couple examples I'll go into are some from the published Murfish literature coming from Xiaowei Zhuang's lab. The first example is this 2018 Science publication where they mapped out the cellular organization, I guess cellular, molecular, and functional organization of the MPOA region of the mouse brain. So this is a brain region that's responsible for different mouse behaviors like mating, aggression, thirst. And from this study, they wanted to understand how the cellular composition of this region of the brain contributes to these different behaviors. So first, they just wanted to map out the spatial organization of this region of the brain. And to do so, they performed Murfish measurements across the region. So in all, they measured about 1.5 million cells. About 500,000 of them are in the control case. But then as you can see from this T-SNE plot, they're able to get very rich cell type identification. So here, they're able to identify all of the key cell types across the brain. And by performing single cell sequencing in parallel, they're able to show that this Murfish cell types correspond very nicely with the single cell RNA sequencing cell types. But as you can see from the T-SNE, there's a richer cellular differentiation that we're able to identify with Murfish. In particular, we're able to identify different subcell types of inhibitory and excitatory neurons. As you see on the bottom left, there's even cases where Murfish is able to more finely differentiate these inhibitory neurons than with single cell RNA sequencing. For example, there's these two clusters towards the end of the plot that correspond with a single-cell RNA sequencing cluster, but two Murfish clusters. So these two Murfish clusters are defined by the expression of neuropeptides, which are very lowly expressed genes that also very functionally relevant in this region of the mouse brain. So since Murfish has this high detection efficiency, we're able to characterize these neuropeptides and the different cell types that they define in a way that's hidden to single-cell RNA sequencing type of analysis. So from profiling these 155 genes across this region of the mouse hypothalamus, we're able to identify major cell types across the brain. In addition to discovering novel cell types not found with single-cell RNA sequencing, and in this way, we're able to map out the cellular organization across the tissue. But in addition to mapping the cellular organization, they were able to layer this on top of a functional map. So by measuring CFOS, which is an early activity gene, they're able to see which neurons were activated during different behaviors, for example, parenting, aggression, or mating. So by looking at the CFOS expression across the different cell types that were identified, were able to determine which neurons or which types of neurons were activated during these different behaviors to map out the functional significance of this cellular atlas. So from here, by combining this functional map with this cellular atlas, they're able to really start to understand how this region of the brain is built and how it starts to impact or contribute to the different behaviors that rely on this region of the brain. The other published example that I want to go into is this bioRxiv preprint that was posted in December. So in this example, they mapped out, they looked at this neuronal culture system and then mapped out the organization of the transcriptome across the different portions of the neurons in cell culture. So for example, with traditional technologies, it's very difficult to understand or sort of determine where each transcript is being expressed across the axon or dendrite with single molecule resolution. But with Murfish, since we're directly detecting each individual molecule, we know exactly where it is along the axon and dendrite. And in these biological systems, the sort of active transport of different transcripts to different regions of the axons or dendrites has very important functional significance, because different transcripts are actively transported so that they can be very quickly translated in different regions in order to create the proteins that are necessary for example, for synapse functions. So by mapping out the spatial organization of the transcriptome across these cultured neurons, they were able to identify sort of groups of cells, groups of transcripts that were similarly differentially expressed among different portions of the axons and dendrites. So both these last two I just went through very briefly because they are published, so you can read more about them if you're interested in either of these applications. But within Vizgen, we have been working to demonstrate Murfish on a very broad variety of different mouse and human tissues. So for many of these tissues, we find that there's very few adaptations to the protocol that are needed. Mainly, we just need to make sure that this sample that we want to profile has good RNA integrity. And then from there, we're able to typically perform a Murfish measurement. So in all, we've done more than 20 different tissue types covering a variety of mouse and human tissues. And we're just now starting to explore additional model organisms as well. So with that, I do want to reemphasize how excited we are to launch the Merscope platform this summer and start to get this research tool in the hands of more researchers, so we can start to really see what researchers are able to do or discover with the power of Merscope in their labs. So with that, I can start to take questions. I think one came up.
Thank you so much, George, for this wonderful talk. This is really an amazing technology. And so before everyone starts writing questions in the chat, I have some questions to start. So my first question is about the QC sample. You were telling about the RNA quality. So how do you do the quality control for that? Because, for example, in spatial transcriptomics that we are doing in the lab, we do one section, we extract RNA to see the quality of the RNA. How do you do for the Merscope?
Yeah, so it's very similar for Merscope. With Merscope, a lot of the RNA quality within a tissue block it is in a lot of cases consistent across the tissue block. So there, just as you do currently, you can take a couple extra slices and then put them through a RIN measurement in order to quantify how much of the RNA is intact. We find that the RIN score does correspond pretty nicely with the quality of the Murfish data that you get as the output. So that's one method just to qualify a tissue block. In addition, we have a verification reagent kit that is single molecule FISH probes that allow you to very quickly assess both the RNA quality, but also how well your protocol is working on your specific tissue sample to see whether there's certain autofluorescence background that remains that needs to be mitigated, for example. So there, with a single molecule FISH probe, it allows you to very quickly look at that to make sure that not just your sample and RNA quality is good, but also that everything else in the process is working properly.
Yeah, and I will say this will be a better option because sometimes, of course, extracting RNA from a tissue, you are extracting bulk RNA, but sometimes the section itself, some part of the tissue is like not well preserved, while the other, it will give you also, correct me if I'm wrong, a better idea of the different regions of the tissue and the RNA quality all over, right?
Yeah, because I think especially for certain human samples where like the freezing process is not always necessarily ideal, or maybe there's even cases where it's biologically induced RNA degradation if it's some tumor where some parts of the tumor have started dying. But there we do see that across these different samples there is a little bit of variation in quality. So this single molecule FISH measurement does give you some indication of that.
And other question, Luciano, tell me if you have questions.
I always had for these guys. So
Yeah, you go.
I guess for me, I don't know, there haven't been, at the moment, there are not a lot of technologies that can do what you guys do, like, just essentially being like single cell resolution or even intracellular, if you like. So my question is, we always look at the single cell, we dissociate the cell, we look at the spatial resolution, the cells get all mixed up, we do the analysis, and then we just classify cells based on clusters, and then we give an identity to those clusters based on the markers that they express. But then when you look at your slide, we're just looking at your slide and the slide in which you can see how all the immune cells distribute across the tissue. You know that they are close to some regions that have more irrigation than others. They are connected to other cells. Have you seen whether that spatial organization can be if you think that this spatial organization has some contribution to the way that the cells would cluster using the standard bioinformatics tools, or another way to say is, are the bioinformatics tool really doing the justice for what the cells really are based on where they are located?
I think that's one of the exciting open areas as people start to adopt spatial single cell resolution transcriptomics technologies, because there's always this question of what is a cell type and what is a cell state. for example, in different regions of the brain, as shown in a more recent bioarchive that I think actually made it to a publication in Nature a couple weeks ago. They, in addition to characterizing the standard cell types, they also saw there were parts of the brain where there were these spatial gradients in the expression of different genes, where when you just characterize it by cell type clustering, you do get these distinct clusters, but then if you project it along this axis where you see this gradient of variation, you can see that it's not quite really distinct, clearly defined cell types that you're identifying. Rather, you're just aggregating this continuous variation in gene expression into these distinct, sort of artificially grouping it into these distinct pieces. So I think there, in that case, I'd say that the traditional single cell clustering approach isn't doing it justice. But also I'd say that there's not a clear tool available yet that's able to pull out these, I guess, very systematically pull out these different patterns and spatial gene expression. I think that's also one of the reasons why we wanted to release this mouse brain data set to help inspire the community to start building up some of the new tools that are required for this new type of data.
Yeah, that's great. That's great. So I guess that the next question for me is, there will be a lot of people excited about this technology to be able to use it and get access as soon as possible. I'm one of those. I'm sure the people around the world is the same at the moment.
Me too.
So, so what is your game plan for the next year or so in terms of where are you planning to home those instruments? Because, I'm sure that there's a lot of logistic plan to where you're going to put those instruments, how to transport those instruments to to Katja's home, in Australia. So how do you plan that? What's the time frame? So we can, it's good to have expectations clear.
Yeah, so our first instruments are planned to go out this summer. So I think that's our next big milestone. We have sort of very, very early, the very first ones already starting to trickle out, but then the pace will start increasing towards as we continue through the summer. But this, as you're mentioning, as you're alluding to, will be mainly focused on North America to begin with, just so that we can make sure that these initial customers that we have are able to have the best support that with this new instrument. If any issues arise, we both keep them sort of in the North America area, so it's easy for us, our field service team, to be able to get to the instrument in case anything is needed. Then we also keep the number fairly limited so that our field service team, our growing field service team, isn't overwhelmed. But as we get more comfortable with the instrument, with these initial users, we do plan to expand the sales of the instrument as necessary, both increasing the capacity within North America, but also sort of expanding beyond North America. So going beyond North America will come in the not immediate future, but I'd say very near future, since it is, we do see there's lots of interest both in Europe and in Australia, and we're very interested in getting our instrument into the hands of those researchers.
Yeah, we're currently working on our infrastructure to be able to enable non-US sites, so Europe and Australia. But what you'll see soon will be our Merscope platforms will be popping up on both coasts of North America and then expanding. So if you were looking to run Murfish measurements, there'll probably be an institution near you that will be able to enable some of those.
Okay, I was going to ask that, whether there will be some sort of service center or provider that you know, like a place that you trust. I guess it will be anyone that has the Murf, you know, that to provide that to make justice of the utility of the instrument, to be able to, you know, channel countries like Europe, Australia, you know, Asia, and getting samples profiled, because otherwise, you know, we as Katya and I are democratizing the knowledge, democratizing all the tech. So otherwise it will be always North America being able to do the cool science, and you know, I'm pretty sure that, you know, we want the other part of the world to have the same possibility, right?
Yes, we've been hearing that loud and clear. So we're working on a path to do that in Europe and also in Australia, in making sure that we have the right infrastructure so that everybody's successful. But it's definitely one of our goals going into 2022, and what that looks like is still on our roadmap for development. But for North America, I think we're really excited. As George said, there's, you know, instruments that are being slowly trickled out shortly. You'll hear some things coming out soon on that. So we're really excited about that. And then in terms of working with providers, that's also in our plans probably for 2022 to help enable access to our technology, right? We're mighty, but, you know, small still. So we're still expanding. So I think having more access to different sites will enable people to get our access, you know, sooner. So
I think it's, you know, it's just about excitement. People is excited to see, you know, and you know, you see all these presentations, the things that you can do, the things that you can see. There are other spatial transcriptomics technology, which, you know, they have their niche and they are really good at what they do. But you know, it's good to have a little bit of competition and a little bit.
And one of the great things about our technology, what George was mentioning, is that, you know, we don't just support human and mouse. If we have a well-annotated transcriptome, we can develop encoding probe sets for different species, potentially like rat and non-human primate. So then it's just working to make sure and optimizing your sample prep protocols to making it successful. And we have different protocols to enable that optimization and before investing in the full encoding probe set. So I think that's one of the unique features of this that will allow it. So for different clinical trials, if you're working with different species like pig or non-human primate, we haven't necessarily done that in-house yet, but there are ways that we can try and explore those projects. So I think that's one of the great the flexibility of the platform and the technology.
Yeah, this takes me to other question that I had, like each panel of genes, for how many samples? Like if I buy a panel of 200 genes, how many samples will I be able to work with?
So we typically sell the panels in aliquots for 20 samples.
Okay. And each experiment is one section that I can do, right?
We define the experiment as one cover slip going into the system.
And about the protocol, since I start, like I said, put the section on the slide or cover slip, and I just go until I get like the results. How much is this time? Like how long is?
So on the bench, it's a few days of time, but a lot of those are spent doing these incubations, hybridizing on these encoding probes, doing the clearing step. So it's a few days, but with a couple hours, just a couple hours of hands-on time. And then it's a day on the instrument, and then a day of image processing analysis.
Let's say 1 week.
I think a week is fair. Maybe 5 weeks.
Yeah,
you can do your sample prep protocol on multiple samples.
Right. So samples can go through the instrument continuously. So in 1 week, you're still able to get 5 or 7 samples worth of data.
Okay. Yeah. Thank you.
And you can batch your sample prep together. So we recommend about a certain duration in the fridge, but you can do multiple sample slides at one time so you can optimize and then feed the instrument continuously. So there are ways that we can work with you on that.
Okay. Thank you. Luciano, do you have some other question? I finished mine.
I have a lot of questions, but I don't want to. I can probably email these guys, otherwise we're gonna make this for 2 hours, and people probably want to go, get
Yes.
dinner or lunch or breakfast, whatever they are.
And we do encourage you to reach out to us if you have a specific project in mind that you wanted to kind of discuss technical details about how would we do projects. We can have those individual conversations, and you can either email us through the info@Vizgen, and then we can reach back out to you and set up meetings, or we can email, tweet us too. We're happy to continue those conversations and discuss projects.
Okay.
All right, fantastic, Guy. Thank you so much for your time and for preparing this wonderful talk.
Oh, Sean had a question. Did you see in the chat, George?
Yeah, there is one question coming just right now.
Good.
Yeah, I think there are ways we can construct probes that can help detect gene fusions. Those are sort of custom type of applications, but we have the capacity, depending on the gene fusion, there's different approaches that we might take.
He's typing. Okay.
Now we can finish. I will say.
All right, guys. Thank you so much.
Thank you so much.